Okay, let's get started. Hello, my name is Alan Hudson from the Immuno-Oncology Translational Network Data Management and Resource Center. I also serve as Chair of Biostatistics and Bioinformatics at Roswell Park Comprehensive Cancer Center. Before we start the seminar, I just have a few bookkeeping items to go over. First, questions will be responded to at the end of the presentation. Please enter your question in the Zoom question and answer box, not the chat box, the Q&A box. The audience can prioritize the order of the questions by clicking on the thumbs up button. Closed captioning is available. The closed captioning for today's webinar can be accessed by clicking on the live transcript option in the Zoom menu at the bottom of your screen. Instructions will also be provided in the chat box. If you're having technical issues, you can make a comment in the Zoom chat box. With that, I would like to turn the proceedings over to our moderator, Mr. John Herndon, who is a scientific lead for the HTAN characterization / biospecimen unit housed within the Laboratory of Spatial and Quantitative Tumor Immunology at Washington University School of Medicine in St. Louis. John?
Thank you very much, Alan. It is my very distinct privilege today to introduce my friend and colleague, Dr. Martha Shrubsole, who is Professor of Medicine at Vanderbilt University Medical Center, where she leads a research portfolio of molecular, nutritional, and interventional epidemiology. A major focus of Dr. Shrubsole's research is to understand the etiology of gastrointestinal neoplasia. She seeks to identify and evaluate modifiable factors, biomarkers, and molecular mechanisms for the prevention, early detection, and precision-based interception of cancer and its precursor lesions, with emphasis on sessile serrated polyps and conventional colorectal adenomas. Some of these areas of study include nutrients such as 1-carbon metabolism, inflammatory markers, gut microbiome, molecular landscape of colorectal polyps, and predictors of metachronous adenomas, including in the NCI Cancer Moonshot Human Tumor Atlas Network. She is also PI of the Southern Community Cohort Study and the newly funded Southern Environmental Health Study, which are two large NCI-funded cohort studies seeking to understand and ameliorate cancer health disparities in the U.S. South. In addition, Dr. Shrubsole has had a leading role in multiple randomized trials and other large-scale epidemiologic studies based here in the U.S. and globally. So it's my distinct honor to introduce Martha today, and her title of her talk is Integrative Single-Cell Atlas of Host and Microenvironment in Colorectal Neoplasia Transformation. Martha.
Thank you, John, for the introduction. And it is also, one of the benefits of these types of programs is that you do get to interact and make a lot of new colleagues at other institutions and learn from people as well. So thank you. Okay, give me just a moment to get this set up a little bit more. Okay, so today I'm pleased to be able to share with you on behalf of the Vanderbilt Pre-Cancer Atlas that is part of the Human Tumor Atlas Network, the work that we have been doing to try to understand the relationship of precancers and how they might progress to become cancer. I don't do this alone. Again, one of the benefits of the Moonshot program is that it's able to bring together people from a lot of disciplines to work on a common problem. And I am honored to be able to lead this along with my colleagues, Bob Coffey, who is a physician scientist, and Ken Lau, who's a systems biologist. So why do we care about colorectal cancer? You don't need to be an epidemiologist to look at this figure and see that the red and blue lines are going down over time. And that is the good news. We have seen a decrease in incidence in colorectal cancer. We've also seen a decrease in mortality of colorectal cancer. And this is probably largely ascribed to polypectomy, removing those precancerous lesions by the implementation of wider and more access to colorectal cancer screening modalities. We see something similar around the world in other countries where there are national programs that have been implemented to screen for colorectal cancer. But that's not the whole story. In this year, we are expected to have almost 150,000 Americans will be newly diagnosed with colorectal cancer, and an additional almost 53,000 will die from the disease. Around the world, almost 2 million individuals will be diagnosed with colorectal cancer. So clearly, this is something that is still impacting a large number of people. We also have started to see that there are groups that aren't seeing the gains, at least not in the same way, or may actually even be increasing in incidence, particularly thinking about those with so-called early onset colorectal cancer that are being diagnosed before the age of screening would normally start. So we do have more work to try to understand what are the causes of cancer, how does it progress, and how can we stop it? One of the best ways that we've had to understand colorectal cancer is to have this, what's called the adenoma carcinoma sequence, this model that we've been using for over 30 years, also called the Vogelgram. And this has been very helpful as we start to characterize what might be some of the steps along that pathway to colorectal cancer. So you can see that on the left, we start with a normal epithelium, and then through an accumulation of changes, either through somatic mutations or through epigenetic changes, we progress to small polyps, to larger polyps, more advanced, and ultimately, if things don't change, we'll end up with a colorectal cancer. As we've gone through time, we have had more techniques and more ability to be able to characterize colorectal cancers. We've come to see that there are primarily two different types of colorectal cancer, two main groupings, although there's certainly other ways to classify them. The first is the chromosomal unstable pathway, or colorectal cancers, which is about 65 to 80, 85% of sporadic colorectal cancers. And then we have another group that are just behave differently and have distinct features that represent about 10 to 35% of colorectal cancers. And these are called CpG island methylator phenotypes or microsatellite unstable high. That's what these cancers tend to be. So given that we know that these cancers have different features, different behaviors, different characteristics, what does that say about the precancers that come before them? And the question would then, next logical question is, do they have the same precursor lesions? And we have come to understand over time that that is likely and almost certainly not the case. So that we have in our first pathway to these the SYN pathway, the conventional or traditional adenoma, which are polyps that we have recognized as precancers for lesions for decades. In addition, we have a newer, more newly recognized polyp called the sessile serrated polyp, also called the sessile serrated adenoma, also called the sessile serrated lesion, that has been more recently recognized as a specific type of polyp that has the ability to progress to cancer. Now, these sessile serrated polyps, we only obtained a consensus diagnosis for them in 2010. So certainly they existed long before the consensus diagnosis. They were even being diagnosed before the consensus diagnosis. But because we have only about a decade of true consensus on what is a sessile serrated polyp, and of course that's even changed a little bit over time, we know a lot less about these particular precursors than we do for conventional adenomas. In addition, one of the reasons that we're concerned about these particular types of polyps is they're a little bit harder to detect on colonoscopy. They tend to be flat, may have a mucus cap. Now, endoscopic practice, fortunately, is adjusting so that gastroenterologists are better able to look for and find these, but they still may be easier to miss than, say, a conventional pedunculated polyp, such as the conventional adenoma. In addition, we see that MSI high cancers, although they make up only 10 to 35% of all colorectal cancers, they're overrepresented in interval cancers. And these are cancers that occur between having a polyp removed and your next surveillance colonoscopy. That could be due to the fact that these lesions tend to be missed, but it might also be due to the fact that these could potentially be a type of polyp that at least some of them may have a faster growth potential and a greater speed with which they progress from precancer to cancer. And so it's important to be able to understand what may be similarities and differences between these polyp types. Sessile serrated polyps are not the only type of serrated polyp that we have. There's also in the green on your far right, traditional serrated adenomas. These are very rare, less than 1%. And so I will not be talking or addressing about those in today's talk, but they are important precursors as well. And then we have the sessile serrated lesions, which I already told you a little bit about. They also tend to be more rare than adenomas. So they're about 5 to 15% in prevalence. And they tend to be predominantly right-sided, although they can occur throughout the colorectum. And then I want to point your attention to these blue polyps, hyperplastic polyps. Now, hyperplastic polyps, unlike sessile serrated, have been recognized for a very long time. In fact, for decades even, they were considered to have pretty much no potential to progress to cancer. That's changing a bit. It looks like there's a very good chance that some of these hyperplastic polyps are actually the precursors to sessile serrated and to traditional serrated adenomas. And there are two main classifications for these: the microvesicular hyperplastic polyp, which is probably the precursor for the sessile serrated lesions, and the goblet cell rich hyperplastic polyp. And so we'll see a little bit of that today. So I told you we know all this information about these different types of polyps. I put up this, fancy diagram about here are the steps we go from normal to precancer. Why do we need an atlas, and what else do we need to do? Well, we still have a lot of challenges ahead of us. For one thing, we have need for better risk stratification. We don't really have good ways to separate out who's at risk versus who's not at risk for progression to cancer. We use histologic features, but we really haven't been able to improve on that. Who could come back, who needs to come back sooner, who can wait to have a colonoscopy or surveillance procedure until later. We also need improved screening modalities. Sessile serrated lesions, even when they're large, tend not to produce blood or release blood. And so they aren't picked up on FOBT or FIT tests, which are the predominant first stage of colorectal cancer screening around the world. We need better early detection strategies as well for both types of polyps. Again, with knowing that one of the best ways to impact mortality, the first step is to prevent people from getting cancer. But if people are going to develop it, we want to be able to detect it early so that it's intervenable and curable. And then finally, we want to be able to identify prevention or interception strategies, including such strategies as chemoprevention, in order to be able to, again, stop cancer in its track, colorectal cancer in its tracks. And so we propose and hypothesize that one of the best ways that we can eventually inform those goals is to be able to better define the molecular phenotypes of precancerous lesions, and to be able to use that information to further understand how those polyps may progress. It is into this that the Human Tumor Atlas Network stepped. Half of the network is focused on precancer atlases, just like our project. And in addition to being tasked with building these multi-dimensional atlases of the microenvironment as well as the lesions themselves, it's also to be built upon data that is prospective, that we have well annotated clinical biospecimens, that we represent all of us in the data that we produce, and that we're able to look at the transition from premalignant to malignant cancer. And we do that through our project that we call the Colorectal Molecular Atlas Project, or ColonMap. And in this project, our goal is to map the spatial and temporal relationships across the spectrum of normal colon. Let me just get my pointer ready here. So this across the normal colon, all the way through early lesions to later lesions, and then ultimately onto adenocarcinomas, including the ability to characterize their unique stromal and microenvironments in order to identify these unique molecular phenotypes that are maybe specific to polyp types, but also may help differentiate within the polyp types for the development of eventual precision diagnostics and preventive strategies. And obviously it takes a lot of people to do something as lofty and as detailed and as comprehensive as this. And so these are just a few of the names of the people that help provide leadership within the program. And I want to point out again here that it really takes multidisciplinary work to do this. You'll see pathology represented, there's epidemiology, there's bioinformatics, and digital histology, a lot of different expertise. And the Moonshot program has been key to be able to bring that together. We can't do that to this scale under normal funding mechanisms. One of the key things that we're doing then in our colon map is to be able to characterize deeply conventional adenomas and cell serrated polyps, so that again we can look at similarities and differences that will hopefully one day be useful for developing new methods of screening, early detection, and treatment. The most important part of our colon map are our study participants. None of this work, none of this understanding and knowledge and gains that may be made can be done without their participation. Individuals end up in colon map by first being scheduled for a colonoscopy at Vanderbilt as part of their normal routine healthcare. And because they're getting a colonoscopy, they're also the lucky winner of an invitation letter from us and phone call to tell them about the project and to ascertain if they'd be willing to participate. For those individuals who do participate, it is a significant contribution that they are making. They complete multiple surveys that ask questions about risk factors related to colorectal cancer in general. They ask questions that are relevant for understanding things that may affect the biospecimens that we're collecting. And so they do spend time answering and responding to multiple surveys. In addition, we mail them a kit, and before their colonoscopy, while they are at home, they collect both a stool and urine sample that they then bring to them with them on the day of their colonoscopy. At the colonoscopy, we collect blood, and then during the procedure, normal colonoscopy takes place. The gastroenterologist will remove any polyps that are found, just like they would if it was whatever type of colonoscopy you would normally have. In addition, they are collecting some normal colon biopsies. So we get colon biopsies from the ascending colon and the descending colon, and that really is going to be able to help us look at a variety of different things, including some of the results that I'll show you today as comparisons to the polyps. But one of the unique things about colon map compared to some previous studies is that we are able to take half of the polyp solely for research purposes. And so we bisect the polyps that come out, and half of that is going to be going to formalin fixed and paraffin embedded, and it goes for the clinical diagnosis. So that is being used to understand what type of polyp it is and to be able to inform the care for that individual. We then take those, once the diagnostics are completed, we then take those samples and bank them and have those available to be used for both imaging studies as well as future genomic and other activities. The fresh half that we take, the research half that we take, is either used immediately for single-cell RNA-seq, which is what you'll see today, or we preserve it for use for future assays. So a few more orientation and context to provide before we look at the results. And the first is to remind everyone that our colons have architecture. So if we look here at this picture, you'll see the lumen area of the colon. So this is the area, this is where your stool passes through, it's through the lumen. And then we have a mucus layer with an outer mucus layer and an inner mucus layer. The epithelium of our colon is then organized ideally in these nicely organized colon crypts. And we'll be seeing several of those today. So I want to make sure that you kind of remember what that shape should look like. We also have within our colons, within the lumen, as well as to some extent within mucus, exposure to a variety of different microbes. Some of them that are there doing very important work, such as helping us to metabolize the foods that we eat, to make the nutrients and the nutrition available to us. So in addition to the architecture looking sort of from the lumen down through the crypt, let's take a look also at the crypt. And the crypt also has architecture. And so what we see there is that we have this base of the crypt, we have stem cells. These are cells that will eventually become differentiated. And at the top of the crypt, we have differentiated cells. These are the grown-up cells, if you will. They're pretty much committed to being who they are, and they don't usually change under normal circumstances. And so please remember that that's something as we look at these pictures to come up, that there's a difference between the top of the crypt, which is exposed to the luminal surface versus the base of the crypt. Now, this is a model of GI tumorigenesis that we understand exists. So this is a stem tumorigenic state model in which something happens to these stem cells at the base of the crypt, for example, biallelic APC mutation, and that causes the precancers and then eventually the cancers to grow. So it sort of comes from the bottom and grows up. We also have another model within GI cancers, but this comes from the stomach. And this is a metaplastic model. So in the context of H. pylori, these differentiated cells can actually transition into a regenerative state of a non-native cell state. And so in normal circumstances, they would not change, but because of this exposure, they experience this metaplasia and reaction, and this is what happens. So as we think about how to do this atlas, what are we trying to understand? These are two of the first key questions that we had and still have, but that I will show you some of the information that we've been able to ascertain in today's talk. And the first one is, can we find evidence of a different model than that stem model for the tumorigenesis within these precancerous lesions? And then secondly, following on that, do these tumors exhibit a different immune tone and microenvironment? So now we get to some of the fun stuff, some of the results. This is our single cell RNA-seq results, and these were done by Bob Chen, who's a graduate student in Ken Lau's lab. So let me tell you just an orientation. I know many will know what single cell RNA-seq is, but let me explain a little bit how it's different from what we would call bulk RNA-seq. So single cell RNA-seq is a newer technology where we're able to look at individual cells and the expression within those cells. And historically, in bulk RNA-seq, we look at it within the whole sample. So you're looking at across cells, whereas this is more closely within cells. And so the first thing that we do here is that we've got a UMAP. Each of these dots represent cells that are in our samples. And these samples include normal colon, colorectal cancer, serrated polyps, as well as adenomas. And we saw in this that we identified several different types of the 9 cell populations, to be exact, of different types of cells within these samples that we analyzed. And I want to point out 3 that are very important for today's talk. First of all is this purplish region here, which are the stem cells. And so these are the cells that I told you are typically in the base of the crypt, and we see those, and those are present across multiple cell types. And then we see these sort of yellowish color in the corner, and those cells, if we look over here at our histology to see what it matches up to, those cells match up to being from conventional adenomas. And so we'll call those adenoma-specific cells or adenoma cells. In addition, we have this pink region over here, which are specific to serrated polyps. And so we will call those serrated specific cells or serrated cells throughout the talk. And please keep this in mind as you see the results, because this picture will come back a little bit later. I also want to point out, using this bottom figure here, that although we talk about these adenoma-specific cells and the serrated-specific cells, just to point out that each of these precancerous lesions do contain normal cells, in addition to histologically normal cells, in addition to having these abnormal cells. We repeated the single cell RNA-seq on another set of data to see if we would see the same thing. And this is just to point out that we do. So this about doubled our sample size. We still see these conventional adenoma specific cells and serrated specific cells when we expand our sample size. And so this is really, I would think, probably the largest collection of single cell RNA-seq data on colorectal polyps. So the next thing that we did was to try to identify gene programs and pathways that are activated compared to normal. And looking at our adenoma-specific cells, which follow this column that has the dark red here, you can see that they have a pattern that is sort of uniquely their own. And what we saw there was that there definitely was Wnt pathway activation, as we would expect from these types of polyps. And they had a transcriptomic profile that was actually quite similar to normal stem cells. However, the stems were also greater in the adenoma-specific cells than they were in the normal from the same individual. Conversely, when we look at the serrated specific cells, we see something different. So now we see a lack, and they follow this column with the very dark colors right here, for those of you who are looking at the figure. They are now actually, in fact, transcriptomically, they appear to look more similar and have differentiated cell characteristics such as KRT20, and they had, in fact, very low inferred stem potential. In addition, they were highly enriched for genes that were expressed in the gastric mucosa, and that's the stomach. You know, here we are in the colon. These are highly expressed in the stomach, and we're seeing them in the colon, including such things as MUC6, MUC17, TFF1, and others, including MUC5AC. So this led us to hypothesize that metaplasia may actually underlie the pathogenesis of sessile serrated lesions. So let's again look at the expression in this UMAP. So remember where those pink cells were located and when those yellow cells were located. And this is showing us the expression of MUC5AC, that marker that we're using for metaplasia. And again, you can see that most of that MUC5AC is most strongly and most consistently shown in our serrated specific cells. And we then organized them into coordinated regulation of genes, into pathways or programs. And comparatively looking at these, you can see that the adenoma with the bright dark red in this pathway is showing a lot of stem activity. Whereas the serrated, you can see it's very little. There's almost no red, a little bit of pink. Conversely, when we look at inflammatory pathways, adenoma has very little, but the serrated is all lit up in red. So this shows us that these particular sessile or serrated specific cells, this transcriptomically resemble these differentiated absorptive lineage cells that produce mucins. They were also enriched for these other non-colonic genes that are expressed in other endodermal organs. And the signature for inflammatory response to epithelial damage was very unique to them. So all of this together seems to indicate an active metaplastic program that's in response to the regenerative, immunogenic, and microbiota activity within sessile serrated lesions. So to validate these findings a little bit further, we did do bulk RNA-seq in a separate data set, including some adenomas and sessile serrated lesions. And again, we saw that same gastric metaplasia signature in the single cell data, and we saw the same Wnt-driven stem cell signature present in the adenomas, providing some further evidence that this was not something that was unique to this small data set. So now we have a way to think about this and hypothesize that quite possibly sessile serrated polyps, or serrated polyps in general, develop from a top-down model. So adenomas appear to likely follow that bottom-up model that we're used to seeing with the stem cells at the base of the crypt, but that these serrated polyps may be in response to damage at the luminal surface and follow a top-down model. To provide histologic evidence of this, we map the location of neoplastic cells by both multiplex immunofluorescence as well as multiplex histological imaging. And this work was done by Elliot McKinney. So I've given you here two representative lesions to stand in place for all of the different polyps that we analyzed, and 3 markers. I want to point your attention to the OLFM4, which is a marker for stem cells, and looks first at the adenoma. When we look at the adenoma, what we can see here is that it is located, you see lots of pink. That's the color you're looking for here. That shows us that there is a lot of stem cells throughout this adenoma. However, when you look at the sessile serrated, it should stand out to you that the pink is nearly absent. And so we, again, see very little of that. Conversely, when we look at this metaplasia marker, MUC5AC, we can see in the adenoma that you don't see MUC5AC, but you do see it within the sessile serrated. So again, and then also seeing that it's in each of these crypts. And when we have looked at this, we do see that the majority of abnormal crypts in serrated polyps do express MUC5AC. So it seems to be a pretty homogeneous feature of these serrated polyps. But you could still have within them some normal MUC5AC at the base of the crypt. Okay, so I'm going to show you just a little bit more detail about that. So let's look over here on this side. We now have 2 types of hyperplastic polyps shown. So these are 2 of the other types of serrated polyps, the goblet cell and the microvesicular. Again, our tubular adenoma, we don't see it. The sessile serrated that you just saw, we do see it. And you can see this really shows a nice picture here in our goblet cell, that you can see that the MUC5AC is really at the surface, at the luminal surface of the crypt. And with a hyperplastic, we also see it concentrated there and also going down into the crypt. So this is again suggesting that the cellular origin of these types of polyps is related to the downstream environment because of this exposures there. We also then have looked at this on a TMA in which we have a variety of different cell types. I'm showing you here first the normal, and we can see, and these arrows I pointed here, in some normal, again, we see MUC5AC at the top at the surface, but not necessarily at the, down into the base of the crypt. And so again, showing us that this is, quite possibly gastric metaplasia or damage at the surface, leading to a reparative state. We also looked at this within the ulcerative colitis. So this is a separate and different disease beyond the sporadic colorectal cancer. And we see here that you see a lot more of that, what looks like gastric metaplasia. So again, providing further evidence that this is an important mechanism within the colon. So now we return back to our single cell RNA-seq data and look at the developmental hierarchy of 7 major cell types, which we mapped onto lineage branches. And through this phylogenetic analysis, I want to point out a couple of key things. So first, looking at our adenomas, you can see that our adenoma-specific cells are that yellowish color that you see in this oval, and then the purple are the LGR5 positive stem cells. And so it appears that that these cells tend to cluster with the stem cells, again, providing some additional support for the idea that the origin for adenomas is within stem cells. However, when we look at the sessile serrated polyps, we see something different. And in this case, they are not with the stem cells, but they are instead sharing a cluster with goblet cells. These pink cells here are the serrated specific, and the green are the absorptive cells. So again, telling us a little bit more, confirming this hypothesis that's forming. So taking another look at our favorite UMAP picture, and remember where the cells are, this adenoma specific, the adenoma specific here, the serrated here, and then the stem cells here. So in a velocity analysis, we look to see where the origin of the cells may come from. Now this is in our normal cells. And when we look at the normal cells, the important thing for you to look for here is the direction of the arrow. The color doesn't matter here, just look for the direction of the arrow. So this is our stem cell area right about here. And you can see that for normal tissue, the arrows lead away from the away from the stem cell area. In adenoma, we see something similar. So now we look at our adenoma specific cells, and you can see here's our stem cell area. The arrows are pointing towards the adenoma. So again, kind of confirming, providing a little bit another clue here that they come from stem cells. However, when we look at our serrated, and specifically sessile serrated, again, in this direction, you can see that the arrows here are pointed away towards, and in fact, in different directions from stem cells, again, providing some confirmation that these are likely coming from differentiated cells and not from stem cells. Now, everything that I have told you so far has been focused on the precancerous lesions on our polyps, but we are interested to know, do these features carry across into cancers? And so looking at MSI high cancers, which are, again, the cancer most likely that develop from serrated polyps, we've done a few different things. First, if I point your attention to this heat map, the microsatellite unstable is the second row. The serrated specific is the fourth row. And we see that the MSI high cancer retained some of the metaplasia signature. This is this serrated specific signature here, but also regained some of the stem cell properties that we don't see within those polyps. However, when we look at the UMAP, even though it regains some of those stem cell properties, it is still its own distinct tumor type, separate from, and that's this color here, separate from the microsatellite cancer. So these are not cancers that are, so it doesn't appear that these are necessarily from the same origin. It just appears that they do regain some of that stemness. And then thirdly, we looked at the MSI subtype. And when we did that, it certainly correlated with CMS1, but so did serrated polyps. So they seem to be related to each other. When we look at the Wnt subtype, these are our microsatellite stable, CMS2, and these are where we see the correlation with the adenoma specific cells. So let's take a look at direct comparison now between the MSI high as well as the serrated specific cells. So again, looking at the programs here, we can see for stemness, again, not much to see here in terms of the sessile serrated or serrated specific cells. However, for the MS high, we do see that it begins to regain that. And in fact, when we look at this through imaging, we see that MSI high cancers have a lot of variation in them. So we have regions within the cancer that have high MUC5AC. So there's not very much pink showing up in here, but there is a lot of MUC5AC in this region. And then we have this area of low MUC5AC where we do begin to see that expression of OLFM4. So that again provides some evidence that although this stem may be regaining and there may be some other things going on, it certainly does appear that it shares this history of metaplasia with the serrated polyps. We also looked at bulk RNA-seq data from TCGA and saw that gastric metaplasia was present in CMS1 and 3, but was not present in CMS2. The data are a bit noisy, so we also did spatial profiling on individual cancers that we had as well. And in fact, we found that none of the microsatellite stable cancers that we analyzed were positive for MUC5AC, so it was completely absent. But 13 out of the 14 microsatellite unstable cancers were positive. And in fact, the stem cell program was present in the MSI high, but it was heterogeneous. So it did not regain everything. And it still had elements that looked like this serrated lesion, just as you saw in this figure that I presented. We also looked at it in single cell RNA-seq with the colorectal cancer in cells with MUC5AC and MSLN and lacking expression of CDX2. We're able to distinguish those metaplastic cells from the LGR5 beta-catenin expressing Wnt-driven stem cells. So again, these seem to be somewhat mutually exclusive in terms of their occurrence within the tissue. Again, this provides us a little bit further evidence of what I just said. So this is now looking at our microsatellite stable cancers. And what we can see is that, again, it appears to be stem cell driven. And we actually see in areas where there's increased proliferation, we see the stem cell features present there. So up until this point, I've really been focusing on telling you some of the information and clues that we're learning in response to the first question. But the microenvironment is also another important component that we can look at in some different ways. In fact, we see that the microenvironmental composition differs greatly by polyp type. So we see, this is looking at immune cells for adenomas, serrated polyps, normal tissue, microsatellite stable cancers, microsatellite unstable cancers. And what we can see is that within polyps, most immune cells are increased in comparison to the normal. And that doesn't seem to be very different between subtypes that they're increased. However, cytotoxic immune cytotoxic immune cells, such as CD8 positive T cells, natural killer cells, were increased in our serrated. So you can see here that there's a difference in terms of the adenomas and the serrated. This needs a bigger sample size to confirm this, but it is very suggestive. We also saw an overrepresentation of the T cells in MSI high compared to MSS, with no difference in the CD4 positive cells, suggesting again that there really is a difference in dichotomy between these types of cancers and these types of polyps in the adaptive microenvironment. We also see differences when we look at the single cell RNA-seq data, which we looked at in order to inform some of our multiplex immunofluorescence. And so I'll have you put your attention to those green and pink blobs that you see on the left of the screen. First of all, we saw that serrated polyps had a higher number of total T cells and of CD8 positive cytotoxic T cells. And the ratio of CD8 to CD4 T cells was higher compared to our adenomas. Secondly, looking at the spatial distribution here, so this is our adenoma, this is our serrated polyp. The spatial distribution of CD68 positive macrophages was significantly different between the serrated polyps and the adenoma. When we look at the adenoma, those little green dots is what we're looking for. And you can see that they're distributed throughout the stroma. But in serrated polyps, they're not, we see this concentration at the luminal surface. Looking now at cancers at the same type of thing, we see that the MSI high cancer, which is this bottom panel here, had enrichment for CD8 positive T cells in the MUC5 AC metaplastic region. So this is this region here and looking at the green dots. Whereas the MSS had fewer T cells, but was very homogeneously composed of the stem-like cells. So again, this strengthens the association between the metaplastic origin of serrated polyps and the cytotoxic immune microenvironment, and again implicates suppression as tumor cells gain stem properties. So in summary, that's just a brief taste of that one. We've identified that there are indeed tumor intrinsic differences between conventional and serrated pathways. The Wnt pathway that has been described is very much present in adenomas, but absent within the serrated polyps. It also appears that we see a difference in that top-down, bottom-up models of tumorigenesis, such that our serrated polyps are potentially arising from damage, a metaplastic response to damage happening at the luminal surface of the crypts. And this could be due and progress further through this cytotoxic immune environment that may precede hypermutation, which is not something that I've talked about today. So we still have a couple of years left on this project, and where are we headed with this? We're continuing to do some of the same analyses and larger sample sizes. We're also, one of the things that we're focused on for the next couple of years is to be able to understand what role the microbial environment may play in this, and specifically looking at biofilms and how they may be microbial biofilms and whether they may be a contributor or related to some of these differences that we see. This project cannot be done without the work of many hands. And I'd like to just point out a few to especially acknowledge. None of this work is possible without the kind and uncompensated participation of our pathology and GI medicine departments at Vanderbilt, who both help with the collection as well as with the permission to use these samples. In addition, it's again a leadership team that crosses multiple disciplines and brings together a lot of different people who are fantastic to work with. So I want to thank a few of those whom I haven't mentioned. Kay Washington is a pathologist who helps lead the biospecimen unit with me. Cindy Sears is a wonderful collaborator at Hopkins, who's an expert in microbial biofilms. Yu Shear and Chi Liu lead our data science core and are very integral to being able to present the types of findings that we have today. And with that, I will stop sharing and welcome questions.
Excellent, Martha. Very, very, very comprehensive talk. And we have a lot of excellent questions here, so we'll get right to those. First question. Does your study include individuals with inflammatory bowel disease, and does this fit into your pathway to cancer?
Yeah, so that's a great question. This is specifically focused on sporadic colorectal cancer, and so individuals with IBD are not eligible for this study. But there are efforts to do similar types of atlases within IBD.
Right. In a similar vein, is there a specific polyp or adenoma type that is associated with colitis-associated colon cancer?
Yeah, so again, we're not able to look at that in our study. So there are particular types that may be, or subsets of even some of these, but that is not something that we're able to look at because we don't include inflammatory disease.
Right. Next question, do you plan to continue following patients who progress to the adenomatous stage, but then do not progress to full-blown malignancy at later follow-ups?
Yeah, absolutely. So this is actually very important. And one of the great things about polypectomy is that you can remove polyps and stop them from progressing to cancer. But that doesn't mean that people won't go on to develop polyps and won't go on to develop cancer. So yes, that's a kind of a long answer to say it's a complicated story to look at, but we are definitely following participants just have their normal routine standard of care, whatever they and their doctor decide is their follow-up, whether it's 3 years or 5 years or 10 years. And so we have their permission to be able to collect information about that. And if it happens during the funding period for this project, we are then trying to obtain additional samples from them at that time. So very important question, help us to understand, do that risk stratification that I talked about earlier to understand who should and should not come back and when they should. These are the types of studies we absolutely need to do to answer that.
Excellent. Next question, the word atlas implies that the goal is to identify general common rules and molecular phenotypes pertaining to each type of colorectal cancer. Then why is there a need for precision medicine, and what drives the CRC or colorectal cancer heterogeneity in patients?
Sure. So we aren't, we're focused on precancers. We include some cancers to be able to see if patterns persist. But again, we don't have great ways, to understand who may be at highest risk. If we can get to the point where we do that, we might be able to have some prevention tools that could be used specifically based upon the features of the individual. I forget the wording of the first part, but there are also other important things that we can do on public health measures that we can do, such as addressing issues of nutrition and obesity and things like that. Those are absolutely important. We are collecting information on that to be able to look eventually down the road at how that type of information relates to these phenotypes that we identify. So it's not a, hopefully we never have a solution. It's going to be a whole toolkit of solutions for every type, all cancers, in colorectal specifically for this one.
Excellent. Next question, did you perform longitudinal or patient-specific precancerous to cancerous lesions?
So a little difficult to figure out how to answer that one. So I think I've addressed part of that. I will say that we have included some of our analysis that we're doing on cancer tissue is we're specifically looking for cancers where they may retain a portion of that tumor, may retain either the adenoma or serrated polyp, so that we can then look at ones that are together. As I mentioned, you take out the polyp, we don't see that polyp progress because it's been removed from the colon, but we will try to follow people to see if they develop it.
Excellent. And also, some people have asked where is it possible to search through your single cell RNA-seq data? Is there a public database and analytical tools?
Yeah, so I would, so I think the analytical tools is part of what's being developed within the HTAN, but I would encourage, if you're interested, to go to the HTAN website, which would provide information to how to connect to the Data Coordinating Center.
Excellent. Do quiescent stem cells or quiescent cancer stem cells play a role in your working model of adenoma or serrated tumors?
So here's a question where having the molecular biologist of the group would be probably better able to answer than the epidemiologist. But certainly this is our first pass through, and we are continuing to sort of expand our sample size, as well as to expand the breadth of the tumors that we represent, including cancers, so that we can start to separate out things like that.
Right. Next question, do you think the adenoma cells can differentiate into serrated cells since they are stem-like?
Yeah, very interesting. So, there was at one time a polyp classification that's not used so much anymore called a mixed polyp that had features of serrated and adenoma in it. So it's intriguing, perhaps it could be. Again, kind of my epidemiology perspective here. Some of the risk factors for these types of polyps, like obesity, smoking, may have different strengths of association with these different types of polyps, but are also common across. So for certainly, maybe to be determined.
Yes, yes. All right, what are the implications for prevention of conventional versus serrated adenomas based on the identified molecular and cellular pathway origin differences?
Yeah, I think we're too soon to say how to turn this into, usable information in that way. But the most important thing that people can do to prevent colorectal cancer is to have your colorectal cancer screening done, age-appropriate colorectal cancer screening, because the best tool we have to prevent colorectal cancer right now is actually polypectomy. It's quite effective. Obviously, lifestyle factors are very important as well. So at the moment, there's not necessarily a way to differentiate between these two to say, oh, this is, based upon these molecular phenotypes, this person should have more surveillance, or this person should have this type of prevention. But hopefully down the road, we'll have a bit more information on that, and can take it into models where we can test that, such as organoids or animal models.
Right. Here's a question about some treatments. So more CD8 cytotoxic T cells in MSI high will suggest that checkpoint inhibitors would work best in these type of tumors. Do you have any response to checkpoint inhibitors? Have you seen any?
Very uninformed about treatment, so I'm not qualified to answer that question, unfortunately. But yeah, we'll just move on to the next one. I apologize. That's not an area of my expertise.
Are there differences in prevalence of serrated versus conventional adenomas in early versus late onset colorectal cancer?
So that's a good question. So actually early onset is quite interesting. And I don't think we really understand fully the relationship of polyps and polyp types. I will say that I think I may have mentioned it, maybe not at the beginning, that the early onset is particularly increased for rectal cancer. And a lot of what we're looking at in the later onset actually is colon. So there's not a lot of reason yet to suspect that the biology is very different for early onset versus late onset, but that it just might be, early exposures or things that might have accelerated that process. But we're focused on, in this project, because we're using screening age, we're really focused on later onset or average onset.
Okay. Yeah. Do you have a cohort with hereditary polyposis syndrome in your studies?
Yeah, so that relates back. So this we do not. That's not what this project is focused on, because it's focused on the sporadic. So we do not include anyone that has a Lynch syndrome or HNPCC or familial adenomatous polyposis.
Some other atlases probably from the HTAN are really working on those as well. So yeah,
Yes.
we refer to those. Do you know what is believed to be the inflammatory driver for the serrated polyps? Is it metabolite, microbe, nicotine, bile, foods, et cetera?
Yeah, so I think that's very interesting, because that's not only are we looking at what happens, we sort of want to know why. And certainly we think that this microbial exposure is one of the main factors, and that is why we see this metaplastic. So we have some work that we're planning to do to test that a little bit further. But interesting to bring up nicotine. Tobacco exposure is very, very strongly associated with risk of serrated polyps, as well as with MSI high cancer. So certainly there could be, very well be other factors beyond the microbial exposures that are involved.
Excellent. All right. How is colorectal precancer and molecular phenotypes different from other cancers? It's kind of a broad question, but.
Okay. So it is a broad question. So let me see. How are colorectal precancers and cancers different from other cancers? So the precancers are different in that there a lot of cancers that have a precancer, it's not necessarily a polyp. So obviously the fact that it's within the colon, there are differences because you're dealing with a different tissue structure on different environment, right? So we're kind of lucky, if you will, in colorectal cancer, because we're able to find and identify those precancerous lesions. It's harder for other cancers where they exist because they're much more invasive in order to find them. So the biology is certainly different, but there may be programs that could be, similar across cancers. Hopefully down the road, maybe there'll be people that are able to combine the information across all of the 10 centers that are looking at this to be able to answer that more specifically.
And also in a similar vein, is there a good animal model for serrated precancer lesions that you're aware of?
Not an animal model person. My colleagues are. So there are some BRAF models that would tie into that. And there may be other models, but I couldn't tell you the name of the mice.
Looking through some of the other questions here, along with MUC5AC, did you analyze MUC17 expression in your UMPA or in tissue analysis studies to ascertain if it follows a similar or different pattern in the sessile serrated polyps and adenomas?
I am fairly certain we did look at MUC17, but I don't recall what the results are. Our multiplex immunofluorescence panel is, I think, 30 to 40 different markers. I've only just presented a few of them today.
Sure.
John, this is probably the last question. Pick a good one.
All right. Yeah, let's Here's a good question. I think, were you able to identify any potential recurrent sequence variation in any of your samples, or are you able to compare origin of the single cells with a sample via sequence variation / identity? Maybe that was one of the flow diagrams that was being indicated.
Okay, let me see if I can follow that question. I'm not sure I caught all of that, John. Can you repeat that?
So were you able to identify any potential recurrent sequence variation in your samples, or able to compare the origin of single cells with a sample via the sequence variation or identities there?
I think the flow diagrams are a piece of that, and there are some other elements. Other thing that I did not present, but that we're doing is that we are doing whole exome sequencing, and we do have some spatial transcriptomics work ongoing too within these same polyps.
So with that, John, I think this is the record number of questions by far for a Moonshot seminar. So Martha, thank you for that outstanding talk. And John, thank you for going through these questions. For those that didn't get their question answered, we'll try to get those to Martha, and she can attempt to relay an answer to you at some point in the future. With that, I'd like to invite you to attend our next Moonshot seminar, November 18th, by Dr. Paul Sorensen, who will be presenting immunotherapeutic targets and oncogenic dependencies for high-risk pediatric cancers. So thank you to our speaker and our moderator, and that wraps up a successful seminar series.